Bismillahirrahmanirrahim. Welcome to my next video lecture. Today we will study the structure and function of mitochondria, plastids, Golgi Apparatus, lysosomes, centriole, vacuole, and we'll discuss the difference between prokaryotes and eukaryotes. What is mitochondria? Singular, it is known as mitochondrion, and it is the organelle present only in eukaryotes. And the major function of it is also known as powerhouse of cell, and the major function is that it is a site of aerobic respiration. It is also known as major energy production center where ATPs can be synthesized. Now, as you can see in diagram that it is composed of two membranes. The outer membrane is smooth while inner membrane form foldings known as crista. These basically increases the surface area where reaction takes place, the production of ATP where takes place. And mitochondria has its own DNA and ribosomes. What are plastids? There are three major types of plastids: chloroplast, leucoplast, and chromoplast. If we talk about the chloroplast, it is again bounded by two membranes. The outer membrane is smooth and inner membrane form sac-like structures called thylakoids. Here you can see the thylakoids in diagram that these thylakoids are present in fluid filled medium, and that is known as stroma. This chloroplast contains a chlorophyll, which is a green pigment, and some other specific type of pigments are also present in chloroplast, which collectively are responsible for photosynthesis. And what is chromoplast? Chromoplast is actually responsible for bright color in flowers, petals of flowers and fruits. And the bright color of flowers and fruits is due to presence of chromoplast. And what is leucoplast? Leucoplast is a colorless substance and which is present in storage parts of the plants, where it is responsible for storage of starch, proteins, and lipids. What are endoplasmic reticulum? Here you can see in diagram that these are network of interconnected channels which are present between nucleus and plasma membrane, or nuclear envelope and plasma membrane. There are two major types of endoplasmic reticulum: rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum have a rough appearance due to presence of ribosomes on their surface, and so they are associated with protein synthesis. While smooth endoplasmic reticulum, they are free of ribosomes. No any ribosome is present on their surface, so their appearance is smooth. And the major function of smooth endoplasmic reticulum is lipid metabolism and transport of materials, and they can also detoxify the harmful substance that may enter into the cell. What are Golgi apparatus? Camillo Golgi discovered a set of flattened sacs which are known as cisterna, and they are present in cell. Here you can see in diagram that these are actually the foldings. These are present in both plants as well as in animal cells, and the major function of Golgi apparatus or Golgi bodies is that it modify the molecules which move from endoplasmic reticulum, and it pack that substances into small membrane bounded vesicles which are known as Golgi vesicle. And after that these substances can be transported into various locations in the cell, or from outside the cell where they are required. What are lysosomes? Rennie discovered lysosomes, and these are single membrane bounded organelles. They contain digestive enzymes, so their major function is to digest food or waste substances. Here you can see in diagram that uh, these lysosomes can fuse with vacuoles, which contain their target material, and these enzymes can break down these materials into very simple substances. What are centrioles? These are hollow and cylindrical organelles known as centrioles. Here you can see in the diagram that these are triplet microtubules. They arrange in such a way that it form a circular hollow structure. And animals have two centrioles near the nucleus that are collectively known as centrosomes, and they are involved in spindle fiber formation. As you know that the spindle fibers are formed during cell division, where chromosomes can attach on these spindle fibers, and the centrioles are also involved in formation of cilia and flagella. What are vacuoles? Here in diagram you can see that these vacuoles are fluid filled single membrane bounded organelles, and the cells have many small vacuoles in their cytoplasm, but in case of plants all these small vacuoles can fuse together to form a single vacuole which is present in center. What is the major function? As it provides turgidity to the cell, and some unicellular organisms they use contractile vacuoles for elimination of wastes from their body. And what is the major difference between prokaryotes and eukaryotes? In case of eukaryotes, the nucleus is prominent as the nucleus is covered by a membrane or nuclear envelope, while in case of prokaryotes, no any prominent nucleus, no membrane bounded nucleus is present. The DNA only floats in a region in a cytoplasm which is known as nuclide. And in case of eukaryotes, the other organelles they are also membrane bounded, while in case of prokaryotes no membrane bounded organelles are present. And if we talk about the size, the eukaryotic cells on average they are 10 times larger as compared to the prokaryotic cells. And cell wall, in case of eukaryotes the cell wall is made up of cellulose in case of plants, and chitin in fungi. And cell wall in prokaryotes is composed of peptidoglycan. Thank you so much for watching my video.